Well, I think we can get started. So I'm absolutely delighted. Well, first, let me welcome you to the SCI Distinguished Lecture Series, and I'm absolutely delighted to introduce our speaker today, Rommie Amaro. Rommie holds the Distinguished Professorship in Theoretical and Computational Chemistry in the Department of Chemistry and Biochemistry at the University of California, San Diego. She has a very distinguished set of achievements. I'll name just a few. She's a recipient of the NIH New Innovator Award, the Presidential Early Career Award for Scientists and Engineers, the ACS Comp OpenEye Outstanding Junior Faculty Award, the ACS Kavli Foundation Emerging Leader of Chemistry Award, and I can go on. Most recently, she got the ACM Gordon Bell Special Prize for COVID-19 research. This is one of the most distinguished award in the high performance computing arena. Her scientific interests lie at the intersection of computer aided drug discovery and biophysical simulations. A scientific vision revolves around expanding the range and complexity of molecular constituents representing such simulations, the development of novel multi-scale models for elucidating their time-dependent dynamics, and the discovery of novel chemical matter controlling biological functions. Many of you might have seen her research being featured in the New York Times earlier this month, and her work contributions towards understanding the structure, atomic structure of the spike protein of COVID-19 has had a tremendous impact. I've had a chance to work with Rommie wearing many different hats, and I'm always amazed when I hear her talk. So without delaying any further, I'll hand it over to you, Rommie.
Thank you, Manish. I'm so delighted to be here. Thanks for that lovely introduction. And yeah, so my name is Rommie Amaro, and I'm from UC San Diego, and I'm going to tell you all about today our efforts to use what I refer to as computational microscopy to study the SARS-CoV-2 virus. Okay, so some of you may have already seen actually a very nice article by Carl Zimmer. This was one from last year that went to press last October. And we were really excited about this work because for a couple of reasons. You know, one was that it sort of represented what we did so beautifully. The images that the virus that you see in the center was one developed, an atomic level model developed in my group. And also the chance to really, I mean, one of the things about this pandemic has been a chance, you know, really for science has become even more sort of infiltrated, I guess the word, into the lives of people with trying to understand, you know, how the virus is infecting us, as well as to develop, you know, potential therapeutics. So, you know, this is to come into the living rooms of people around the world is really cool. So what I want to do today, I'm going to tell you about our work to develop and simulate these atomic level systems to give you the story behind the pictures. Okay, so much of the work in my group revolves around utilizing all atom molecular dynamics simulations. And I like to refer to this method as a computational microscope. And that's not an analogy that I developed, but one that I stole from my former mentor, Klaus Schulten. And I think it's exactly the right way to think about it, because it's really what we're trying to do is to use computing, high performance computing, as a new type of microscope to give us unseen views into the inner lives of biological and other types of organisms. Okay, and so what we can do nowadays is we can actually combine many disparate sources of experimental data taken in different groups using different techniques, you know, all around the world. So we can combine these different data streams into one highly detailed three-dimensional model of the biological system. And so what this might look like nowadays, we can combine multiple different types of biological structural data that are acquired at different resolutions. So we can combine high resolution X-ray crystallography with single particle cryo-electron microscopy, as well as cryo-electron tomography. And this is all different types of structural data. But then we can, once we build those structures, we can also ingest or incorporate other types of experimental data sets like glycomics, which is really mass spectrometry data, as well as lipidomics and genomics. So we build these highly detailed three-dimensional systems, and then we approximate those systems down to their many atoms. And we essentially treat each of those atoms as hard spheres. And then all we're essentially doing, we define this potential function here, which, depending on what your field is, you may think it looks complicated, but it's actually sort of a really quite simple representation of the different interactions that all the atoms are having with the other atoms in the system. And then all we're doing is integrating Newton's equation of motion over time. So we start with this model that's built on experimental data. And then we're numerically encoding that model. And then we start with a particular configuration at time t=0. We perform this integration with a rather small time step. The time step here is about between 1 and 4 femtoseconds, depending on sort of some of the tricks of the trade. But so that's really short in terms of biological timescales, where we want to, we're mainly interested in maybe things that are happening at the microsecond, millisecond, or seconds or longer timescales. I'll tell you about some ways that we can sort of get over those challenges. But we perform this numerical integration millions and billions and trillions of times. And we are able to build up a dynamical understanding or a trajectory of the time-dependent dynamics of the system. Okay, and so nowadays we compute this integration on this, really fantastic supercomputers. One that our work for the virus relied quite heavily on was this wonderful NSF machine at the Texas Advanced Compute Center called Frontera, which I'm showing here, as well as Summit, which is at the Oak Ridge National Laboratory. These two machines have been, on the top 5 or top 10 of the top 500 list for a number of years, and they are awesome resources. So what I hope to convey to you in my talk today is, really not only how we can use simulation to sort of make these cohesive models that unite many different types of data sets, but then how we can use simulation on top of that to really extend what we can learn from experiment. Okay, and so just a little bit by way of background. So for many years, my group had been studying a number of infectious diseases. We were particularly interested for many years in influenza virus. And, I would say until about February of 2020, we were really consumed with studying influenza. But this mid-February of last year was a really special time for us and many others in the world. So in mid-February, a paper that I had my group had worked on for a number of years. This was a project that involved the construction or building of the all-atom influenza viral membrane, which is shown here on the cover of this journal. We were able to, it took us years, like 6 to 7 years to build the tools and to develop the simulation techniques and analysis tools to build and simulate the influenza viral envelope. And so we were really happy in mid-February after, 6 or 7 years, this work finally saw the light of day, was published in this great journal, ACS Central Science. But that same week, things around the world were really starting to change quite dramatically. So what I'm showing here is a total confirmed COVID-19 cases on log scale around the world for the early months of the pandemic from January through May of 2020. And so like in mid-February, many of you may recall, there weren't really very many cases circulating at all in the United States. There was maybe about a dozen known cases, but we started to already sense a change or a concern, that we knew that this virus had escaped mainland China, and that was already looking particularly grim in the Lombardy region of Italy. So this was around the time that we started to shift our attention to this new threat. And then that same week, something else really important happened, and that was that the group of Jason McClellan, together with collaborators at the National Institutes of Health, deposited into the bioarchive the first cryo-electron microscopy or near atomic resolution structure of the SARS-CoV-2 spike protein. And this was really the date that everything in the group, that things really changed for my group. So I'll never forget when I saw this article, we immediately, I walked down to the offices of some postdocs in my group and a grad student. This is Lorenzo Castellino, Ziad Gayeb, and Abigail Domer. And this group had been working really hard on studying, influenza and other sort of complicated biological systems. And I told them, I said, I think we need to really start to do something with this. And so it picked this up very rapidly. And, we originally were thinking, okay, like, let's set out to do what we just did with the flu, but with this new virus. And then the next thing I did was I came back to my office and I sat down and I contacted Dan Stanzione at the Texas Advanced Compute Center, who's a fantastic director there. And, I told him, I said, hey, there's this virus. And he's like, yeah, Rommie, I know, I've heard about it. And I said, I think we need to do something about it. Maybe, with simulation. And he was just so generous and gracious at the time. And he said, Rommie, I know what you guys do. And I just want to, throw all my support behind you. So, you can get started with the computing and then we'll figure it out, because typically you have to have, really these formal allocations and so forth. But so that really enabled us to be able to sort of move very quickly. So before I get started too far down the technical details of the work itself, let me give you just a little bit of background on the virus. And so by now, I'm sure probably almost everybody is very familiar with the virus. This is an image that was created in my group of what it looks like. So it essentially looks like a golf ball with spikes sticking out of it. And these spikes are in fact called the spike proteins. And, we care about the spike proteins because they sit on the outside of the virus. And so they're the first point of contact that the virus has with human cells. So they play a really key role in the infection process. On top of that, they are also highly antigenic molecules. That means, that your immune system, when faced with this molecule, will create neutralizing antibodies and have an immune response to it. So the spike proteins, in addition to being functionally very important in terms of the infection process, they're also, as many of you probably know and hopefully have been injected by now, they're also the component of all the major approved vaccines. So a key step, so most of what I'll tell you about today involves the beginning steps of infection. And I just will say, and some folks, you may ask questions at the end, I'm not sure. The spike protein itself is a spring-loaded molecular machine, that does a number of, it undergoes a very large conformational transition, sort of linking the host and the host cell membrane and the viral membrane, catalyzing that fusion process. What I'm really going to focus on is the very first step of infection. And so in this first step of infection, the spike protein, which I'm showing here in sort of white color, has to interact with or make contact or find on the surface of the host cell a protein that it will recognize. And so what it recognizes is this yellow protein here called ACE2, or angiotensin converting enzyme 2. And what I'm showing here is this sort of a close-up of this interaction. And so what I really want to bring your attention to is this light blue bit in the middle. This is what we call the receptor binding domain, or the RBD of the spike protein. And that is the part of the spike protein that really makes the important contact with ACE2. It really forms like a handshake almost between the host cell, the human cell, and the virus. And that's really what sort of precipitates the whole infection process. So I'm going to keep coming back to the RBD, and I'll try to keep the coloring in blue throughout. So when we first, the first data that we, were handling was the spike protein. And I'll just say, when one aims to build these big systems, the way that we, and simulate them, the way that we sort of attack that is not to just simply like build the whole system all at once and simulate it, but instead to take more of a reductionist approach. And so what we do is we try to create, for each of the molecular piece parts that feed into the whole system, we try to create the best possible models. And by best possible models, I mean, that they're really keeping pace with, all of the experimental data, and hopefully are even experimentally validated. And then we bring those pieces together into the working whole and simulate that. So the first part of this whole puzzle that we went after was the spike protein, in part because, as I had mentioned, the McClellan group had made this data available. This paper was published in Science in March of 2020, and by the way, has already been cited, I think, some 5,000 times since then. I mean, the impact of this work has been just enormous. And what they presented was what I'm showing here sort of in the middle of the screen. This is the structure of the spike head that we refer to it as a spike head. It has the RBD in this case is shown in green, and it's in what they call an up or an open conformation. And you can see it sort of sticks out above the rest of the spike. A few weeks later, another fantastic structural biology group, the group of David Wiesler at the University of Washington, published a study in Cell that presented another set of structures for the spike protein. These structures largely recapitulated what the McClellan group had found with the one-up conformation of the spike, but they also used symmetry with cryo-EM analysis in order to develop a model of the closed spike. So this has all of those RBDs in what we call a down conformation. And then just one other thing that I should have mentioned probably already, but I just buzzed right past it, was that the spike itself is a trimer, and you'll see the symmetry as I show you more pictures. It's a trimer, and each of those 3 protomers has the end-to-end sequence is about 1,300 residues with different domains sprinkled throughout. So the part that is highlighted here in bold is what they were able to resolve with cryo-EM. Now, as great as the cryo-EM method is, it's not perfect, and it has one of the things that it has trouble resolving is our highly flexible parts of these systems. So in these initial data sets in particular, there were many missing loops and so forth that we needed to model in. So my group used sort of standard molecular modeling methods in order to develop complete models of the spike head in the open or the one up and the all down or closed conformations. But so beyond the head, these spike proteins are connected like a tether to the viral membrane through another set of domains that we refer to as the stalk. And then beyond that, they also have this thing inside. So in pink here is what we call the transmembrane domain. And then inside the virus itself, there's another domain that we refer to as the cytoplasmic tail, shown here in purple, that sort of sits inside the viral membrane. So we again use sort of standard homology modeling approaches to come up with structures for these parts of the protein, which they can't see in cryo-EM because they basically move around too much. So this was our fully built end-to-end structure of the protein itself. But then beyond the protein components, the other thing that the structural biologists, experimental structural biologists, are challenged to see, again, because they move too much, are these things called N and O linked glycans. So N-linked and O-linked glycans are a very important post-translational modification of proteins. They perform all sorts of important biological functions for proteins. They help them fold, they could help them with trafficking, they can do any number of things. And we know that they're there, but we can't see them, or the structural biologists can't take pictures of them because they're so flexible. But what we do have, and what became available, I think in about April or May of 2020, was using mass spectrometry, using glycomics methods. The group of Max Crispin, again together with Jason, were able to determine, using glycomics, the molecular composition of each of these N-linked and O-linked glycans in the spike protein. And so the spike protein, each of those protomers that I was telling you about has 22 N-linked and a handful of O-linked glycans. So the whole composite structure has 60 some odd glycans attached to it. And you can really see how it's basically covering the whole structure of the spike. And so, one of the interesting things about viruses and sort of that we've known for a long time is that one of their sort of mechanisms of infection mechanisms that they've adopted or evolved to have is that they basically cover themselves in these N and O linked glycans. They basically cover their envelope proteins, as the proteins on the outside of the virus, with these glycans in order to essentially hide themselves from the human immune system. Because it turns out that all of our own sort of normal cell extracellular proteins are also glycosylated. And so what happens is that or just the glycans are in general like a part of proteins that is sort of native to humans. So the immune system doesn't recognize that as foreign. Instead of it's a way that the viruses have evolved of basically disguising themselves essentially from the human immune system. So but we can build these molecules back in with these modeling techniques, and we did that. And then the other thing is really, we needed to make a model of the viral envelope. And so we know that the virus buds off the endoplasmic reticulum in the Golgi intermediate compartment. And we know from many, decades of fundamental biological studies that the different membranes in our body, they contain different phospholipid compositions. So we recreated an in silico membrane composition for the virus that's reflective of what it should be actually experimentally. And so you can see the relative proportions, stoichiometric proportions of these different lipids, phospholipids, as well as, for example, cholesterol. And I don't tell you the names here because the names are really long, but you get the sense that there's a handful, about a handful of important lipids, and we've created a model that reflects that. So now what I'm showing you is basically a slice through the viral membrane. So sort of looking at the bottom of the spike protein. So that membrane I was telling you about, we've colored here in pink and red and orange and fuchsia and yellow. These are the different phospholipids I was just telling you about. You can see the transmembrane helices of the spike protein. Those are shown in gray. And I just want to highlight, so there's yet another post-translational modification that is also experimentally very difficult to study, but that we've included here. And that's the cytoplasmic tail has a number of cysteine residues that are palmitoylated. And so these palmitylation groups are these just basically like very greasy aliphatic groups. They look like caterpillars here in this picture. They basically snorkel up into the membrane and help anchor the spike protein in the viral membrane. So, and that's a common sort of thing for these viral proteins to have. So our model also includes those. So now what I'm showing, and hopefully there's not too much of a lag with Zoom, but this is just a movie that shows sort of the whole construction of the system, just to recap. So we have the whole end-to-end protein. We built that model both in the one up conformation as well as the all down conformation. We have all of our N and O linked glycans, and you can see how they just decorate the structure from top to bottom. We have our viral membrane with the appropriate composition, and we also have those palmitoylated cysteines. And so this is what our full system looks like at the end of the day. And I'll also say we do have water molecules and other like buffer ions. So these are fully solvated molecular dynamics simulations. I don't show you the waters because if I put the waters there, you can't see the protein. So we sort of hide those from view, but in the simulations, there are waters there. So we build the system and then we simulate it. And so now what you're seeing is the wiggling and jiggling of the atoms as predicted by the molecular dynamics simulations. And so what you can see hopefully here is, you can sort of watch the movement. And I think it becomes really apparent that the glycans in particular are very mobile. You can see how they just sort of wag around. And that's, just supporting, or gives a supporting reason, as you can clearly see why structural biologists would have a difficult time sort of getting a crisp resolution of those side chain conformations. So we built, again, just to recap, the up state, the down state, and each of these models has about 1.7 million atoms. So, not huge, but pretty big size. We used the Charm36 force field running NAMD2 on the TAC Frontera machines, and we were able to get about 60 nanoseconds per day using 256 nodes of Frontera, which is really terrific benchmarks. And so in relatively short order, we were able to accumulate about 4 microseconds of sampling, dynamical sampling for each system. And then before I move on, I just want to say quickly, I think, for those who are maybe less familiar with molecular dynamic simulations, I just want to say, that these images, they're beautiful and they're striking, but they're more than just pretty pictures. And it's not like a video game. It's these simulations molecular dynamic simulations are numerical statistical mechanics. And so what that means is that, we pay a lot of attention and pay a lot of computing resource price to solve these equations in a way that is theoretically rigorous. So what that allows us to do is to extract back out from averaged microscopic properties experimentally testable macroscopic observables. These simulations can make direct connections to things like binding free energies, heat capacities, viscosities, etc. So that's the real power of this approach. And that's hopefully today I convey to you sort of, why this can be so useful when working hand in hand with experimentalists to test the predictions that come out. Okay. So what did we learn? Whew. So one of the first things that we learned, which was so striking to see, was we showed the world for the first time what the full length, fully glycosylated spike protein actually looked like. And so on the left, what you see is the protein itself shown in blue, and the viral membranes at the bottom. This is essentially what the structural biologists see. They see the protein. On the right, you see how much different it looks if you actually consider the glycans. So on the right, each of these, the dark blue bits, these are the glycans that I was telling you about, those N-only glycans. And so each sort of tuft or little ball of fur that you see there represents a composite image of a snapshot of many snapshots extracted from just one glycan across a microsecond of dynamics. Okay, so like this puff here is like all of the space that's occupied by this particular glycan. And so what you can see is how each of these glycans over the course and the small timescale dynamics sort of sweeps out like a volume of space around the protein, and together they create essentially like a dynamical fur coat almost that basically shields and disguises the protein from our immune system. So this information was, it was striking to see it. But this kind of information is also really critical for potentially for vaccine design as well as for drug discovery. And people are curious not only to know where the glycans are, but where they're not, because places where you don't see glycans, in general, we consider those to be sort of vulnerabilities of the spike protein for reasons of therapeutic design. Okay, so so the other cool thing was that our simulations actually showed why the spike protein adopted these two different conformations, so why it was up and down. And so if you look here, what I'm showing on the left is a top-down view of the closed spike. And the receptor binding domain, that part that is really important that needs to make contact with that host cell receptor, is shown again in light blue. And what you can see is that the glycans sort of cover up, shield part of that receptor binding domain. And here from the side, you can see, this is a side view, so we're just tilting it. You can see how the receptor binding domain is really tucked down beneath those glycans. However, if we looked at it when we just did the same looking at the dynamics and the structures for the open spike, what you see here, now you see how the receptor binding domain, the part is presented completely differently. It's no longer shielded by those glycans. And similarly from the side, it's particularly striking. You can see how the receptor binding domain has now popped up above that shield. And this is, to me, every time I look at this, I still think it's so amazing, because it's this part here, this tiny part, this is like just part of it's really just one part of the receptor binding domain, such a small region of the spike, but that is the part that has to make the contact with ACE2. You hide that, you prevent infection. You show that, you have a real weapon. So so that was interesting. So but beyond shielding, so people kind of have always thought of glycans as being these shielding molecules, because that's sort of really, well, that's really how we've known them to be. But something else interesting popped up, and it got us to wondering if maybe these glycans were doing something beyond shielding. And so so I want to say this work was a wonderful collaboration with Elisa Fada. She's a fantastic glycochemist / glycobiologist at Maynooth University in Ireland. And when we were looking at the structures, we noticed something interesting. And so on the left, I'm showing you, the spike protein, we're looking at the top-down view, and you can see the threefold symmetry. We always noticed always one glycan that was always sort of sticking out into solution. So here, this glycan here is shown in blue and green spheres. But when we looked at the open spike, what we saw, or the spike with that receptor and binding domain and that one-up conformation, we saw something else. We saw two of those glycans were doing what they normally did. They were sticking out into solution, but one of these glycans was actually sticking inwards. And so we took a closer look. And now what I'm showing is sort of a side view of the top of the spike protein. The receptor binding domain I'm keeping in this light blue color, so you can see that here. Red, the other chains of the spike protein, parts of the spike protein are shown in red and gray. And I want to draw your attention to these two glycans here. So what we noticed was that when we built in those glycans that were missing from the cryo-EM data, that we noticed that for two of these glycans at position 165 and 234, these glycans filled up the void space that was created when the RBD moved into that up conformation. And so we thought this was really interesting, and it occurred to us that perhaps these two glycans were acting to structurally support that part of the protein, that RBD, in that up conformation, much like a kickstand on a bicycle. And so we then created an in silico mutation. So we mutated these asparaginins to alanine, and that would eradicate those glycans at those positions and leaving sort of a hole there. And we basically reran the molecular dynamics simulations with this mutant structure in order to test the hypothesis of whether or not these glycans were actually acting to structurally support it. And what we saw in the dynamics, which I don't show you here, but is in the paper, we basically see a great destabilization of that spike of that RBD domain, and it actually sort of collapses downwards. So then we said, okay, well, maybe there's something to this hypothesis. And so, the next thing to do was to try to get it tested experimentally. And so, so on Twitter, and I had just recently joined Twitter. I had never done really any social media before, but at the start of the pandemic, I joined it, and it was just sort of really remarkable for me. This whole, there's like I call it nerd Twitter, but there's this whole like science Twitter verse, and it's a fantastic way to meet colleagues and to keep pace with all of these cutting edge experiments, and has been really important for keeping up to abreast of the developments with SARS-CoV-2 science. I think it's really been phenomenal. So anyway, so I ended up interacting a little bit with Jason McClellan on Twitter. So Jason, you recognize his name because he was the PI on the spike protein structures. And I just emailed him and I said, we have this crazy hypothesis, and here I'm thinking this guy's going to be like so busy because he's the co-inventor of the Moderna vaccine, and he made really seminal contributions to our active vaccines. But anyway, he was very gracious and said, no, yeah, let's do this. Let's test this. So Jason and two folks from his group, Jory and Christy, they created an experimental biolayer interferometry assay, which basically allowed us to test the effects of these mutations on the ability of the spike protein to bind ACE2. And so in green here is the positive, this is our positive control. This is the normal, 2P spike. It's a proline mutated spike. That interacts very well with ACE2. On the bottom in gray is a spike that has a disulfide bridge that basically keeps it locked down in a down conformation. And so this is sort of our negative control. It's always locked down, so it can't ever pop up and interact with ACE2. So, so then you can see here these two intermediate lines. This is the testing of the effects of these two mutants. They're just single point mutants. They didn't get enough protein expressed for the double mutant, but what it showed was indeed there was a marked decrease in the ability of these two glycan mutated strains to interact with ACE2. So, so we were, excited to see this. So that basically supported our hypotheses that these two glycans were actually, doing more than just shielding, but in fact, actually participating in the infection mechanism itself. And that turned out to be something that actually no one had ever seen before for viral proteins. So it's sort of opening a new paradigm for functional relevance of glycans in biological systems, which was really cool. So this paper was published in ACS Central Science last October. And then, so glycans turn out to be so interesting, though. They also are really important to potentially help immune escape. And so in another study with Reno Rapuli and Jason, again, McClelland, they basically incubated the SARS-CoV-2 virus with a highly neutralizing convalescent plasma. So basically the plasma of someone who's been infected with COVID-19 and developed antibodies against it. And what happens when they do this is that over time, the virus keeps mutating and mutating and mutating and will escape. It will escape neutralization by the antibodies. And so after what they saw was after about 3 months of this incubation, the spike protein adopted a very large mutation. I mean, it had a number of mutations, as you can sort of see here, sort of increasing along the way. But there was one large mutation in another part of the spike protein called the N-terminal domain that not only inserted a big loop here, which I'm showing in yellow, but also encoded for a new glycan sequence. So these glycans are actually, you need a 3 amino acid letter sort of sequence that then these enzymes recognize and say, okay, add a glycan here. And so when the virus is mutating over time, it can change the glycosylation profile, and sometimes it changes it and the proteins don't fold right. But other times it changes it and everything works fine. And it could even be advantageous for the virus in terms of fitness. And in this case, it was, because what happened is, I'm showing on the side, this would be on the bottom here is the spike protein. On top in gray is a neutralizing antibody. So in the normal case, the neutralizing antibody would be able to bind onto this part of the spike protein, and that would be one of the ways that it's neutralized by us. But if you think about it, if the glycan, if the virus now has an additional glycan, which is this large sort of blue molecule now sticking out here, that's been introduced right in the middle of where that antibody needs to bind. So it just completely obliterates the ability of any neutralizing antibodies actually that had already been developed to neutralize. And so we've actually seen some of this, some manipulation of the glycan shield already for Delta. Omicron, which I'll talk about in a little bit. Do I talk about that? I talk about that next. Actually doesn't actually change the glycans. But so here's a picture of Omicron. So I have this a little, trying to keep this talk, really up to date. So this is a spike protein, and Omicron, we're still getting data for it, but it's concerning because of the extensive mutations that it has. Oops, I wanted to pause that. So let me see if this will play. So it has a number of mutations. It's significantly more mutated than any strain that we've seen circulate yet. And so all the different mutations here are now shown in these pink spheres. And so you can see, and amazingly, many of those mutations are in the RBD, and actually in that part of the spike protein, this top part here, which needs to bind ACE2. So that's surprising to see, because one ordinarily thinks that if you make too many mutations there, that you'll disrupt the ability of the spike to actually interact with ACE2. But in this case, it retains some function. So is this guy playing? It's getting tired of playing. Okay, I'll just let it play through. Okay, so you have all these mutations in the RBD, but then you have other mutations as well. Here it's showing where those mutations would affect the interactions with ACE2. We've kept that coloring in yellow as from before, and you can see how those mutations are directly at the interface. But somehow, these mutations collectively are still enabling and allowing ACE2 to be recognized. And so, the data is just rolling in now, but it does appear to be really sort of highly transmissible. I think that's at the end. Yeah. So it's still able to retain the function. Okay. And I'll come back to Omicron a little bit more at the end. But so everything I've told you about has really been looking at sort of the short time scale dynamics around 2 different experimentally characterized states, the closed state and the up state, or the open state. which was, you know, useful, right? We discovered that finding about the glycans and so forth, so that was useful. But, you know, what we're really also interested, we really want to know how this thing works. And so, so ideally, we would be able to simulate this opening transition. And in fact, we would do that with MD if we could. Oh, it stopped. I will never understand this movie. Sometimes it runs and sometimes it doesn't. I apologize. So the deal is that in order to sort of study this opening transition, that's a fairly slow biological process. We think from FRET experiments that opening happens on the order of seconds. Okay, so if we go back to the beginning of the talk, you know, if our time step is 1 to 4 femtoseconds, it's going to take us a really long time to get to seconds time scale, like years. So, so we used, instead of using conventional molecular dynamics, we used an enhanced sampling technique called weighted ensemble MD. And this is a really cool approach that basically sort of helps the system get over these interesting functional barriers that otherwise would sort of keep us, you know, down here in this valley where nothing happens. You know, we want to move around on the energy landscape to see what's happening. So this has been a wonderful collaboration, I have to say, with Lillian Chong and now also Dan Zuckerman. But it gives us dramatically increased efficiency for sampling and has allowed us to actually sample this sort of opening of the spike protein. I'm sorry, you can't see the movie here. There'll be another one in a minute. But it allowed us to do that in just a matter of a few weeks' time using the Longhorn supercomputer at Texas. So at the same time, when we were investigating these simulated pathways, it turned out, serendipitously, we discovered that Joachim Frank, who is, you know, Nobel Prize winner for chemistry in 2017 for cryo-EM, had been working with a graduate student, Evan Seitz, to also explore the cryo-EM data set that Jason had made available. And so, you know, with cryo-electron microscopy and most structural biology, you know, they're like hyper-focused, you know, for good reason, but they're like hyper-focused on determining these high resolution structures. And, you know, the thing about that is that you really end up losing a lot of the dynamical information. And so with cryo-EM, they'll have like hundreds of thousands of images, and then they bin the images and they focus on like the dominant set that gives them the high resolution structure. And then they essentially throw away everything else. And there's been a number of, you know, really great groups who've been now trying to see like, well, what else is that data telling us that we can extract out? What kind of data? So Joachim has been working with collaborators in Wisconsin to develop a method called manifold EM, which is an artificial intelligence-based method that is trying to extract out dynamical properties from the images that otherwise would be thrown away. Okay, and so why is she telling us about this? Well, because they had been looking at the same set of data. And so on the left, what I want to show you is we were able to look at these simulated opening pathways using weighted ensemble. And on the left, I'm going to let it play again. On the left, this is what our simulated pathways look like from the simulation. On the two right-hand panels are basically the opening pathways that they see if you actually take a closer look at the cryo-EM data that was being thrown away. And so this allowed us to basically it basically helped us to validate that those predicted opening pathways, when you compare our simulated pathway with the experimental data using this cool method, what you see is that there's a pretty high alignment, a very high alignment along some particular projection directions that they could resolve, basically supporting that, okay, these simulated opening pathways, you know, they look reasonable. Okay, and so so this movie will play. So here, what we were able to discover using this weighted ensemble method, and here you're finally going to get to see this full movie of the spike protein actually going from the closed to the open conformation. And again, here I'm showing the receptor binding domain in this light blue, and the other parts of the spike protein in gray and pink. There it goes, and there it's open. So we went from closed to open. It's going to play again, and I'm going to keep talking. So what I want to draw your attention to is this glycan here at position 343 that's in hot pink. And really what I want to drive home is that when we did our first studies of the conventional MD, and you look at if you look at the closed state dynamics, that glycan that I'm showing in hot pink, it sort of sits down at the side. it doesn't look like it's doing anything interesting. It looks like it's part of the shield. So we didn't pay attention to it. When we look at the receptor binding domain in the up conformation, that guy looks like he's still in the same spot, like nothing interesting happening. But when you look at the actual opening mechanism, what you can see so clearly is that this glycan is actually helping that receptor binding domain to actually open up. And so we actually are calling this now a glycan gate, because it's essentially, at first it's got the gate closed and then it kind of opens. It's interacting with all these different residues along the receptor binding domain, sort of making like a hand drive kind of motion. And then it like a molecular crowbar kind of kicks it out and open, which was super cool. And I think what I just want to emphasize is that like it's like we can learn so much more from these simulations, and it's sort of, I think, a beautiful example of what these advanced sampling techniques can actually get you. And so we went back to Jason. They were kind enough, and his group, and they were kind enough to run more mutations for us. And what they found was that actually this glycan at 343, which we didn't even notice in the first set of simulations, it turns out it's even more important for spike function. It dramatically reduces the ability of the spike to interact with ACE2. And in cell-based assays, but from another group, actually they show that if you mutate this, the virus can't infect cells at all. So that was really cool. That work actually was published in Nature Chemistry, but I didn't update the slide with the citation. Sorry about that. Okay. So then moving beyond the spike, I mean, as I mentioned in the beginning, one of the things that, and I'm just in general really excited about, is in increasing the complexity of the sort of actors that are represented in these simulations, because I think there's so much to be learned from doing that. And so, in late last summer, there were just like, there was like a dominoes set of structural biology papers that came out using cryo-electron tomography, which is, sort of one step up in terms of complexity and size from single particle cryo-EM, where they actually were able to visualize in 3 dimensions the virus like in situ. And this gave us all sorts of really cool information about conformations of the spike protein at lower resolution. It also told us about the average dimension of the viruses. You can see here a couple of these particles recreated, as well as how many spike proteins were on the surface of the protein and their distribution. And so this was really beautiful data for us to do modeling. And so, I just want to say that for simulating at the next scale, which would be the virion scale or the whole virus scale, the single biggest obstacle to doing that is really creating a good model or a numerically stable model for the membrane bilayer. And that's essentially because differences in curvature of the inner and outer leaflets creates a lot of numerical instabilities that need to be worked out. So we and others have been working on this problem for years, and we've developed a tool called Lipid Wrapper, which we used here, which basically allows one to build all atom membranes at any sort of arbitrary geometry that you fill in. And then my amazing graduate student, Abigail Domer, who just defended a couple of days ago, was able to, she developed a workflow that was able to get this virus up and running and stable in sort of its all atom form, fully solvated. So what you're looking at now is the fully constructed virion, where we have the dimensions of the virus and the patterning and density of these spike proteins taken directly from the cryo-ET, and the wiggling and jiggling that you're seeing there is actually the dynamics run on the Summit supercomputer last October. And so this simulation had about 305 million atoms, and really it set a new high watermark for computational virology. So, and to do it so quickly, the studies with the flu took us 6 or 7 years to do. But then once we had built the tools and sort of, and really had the experimental data in hand, we showed how quickly it could be done, which was really cool. So then since last year, we've been working to, as as I mentioned earlier, talking about Omicron, or Omicron, the virus is mutating. And so we can continually update our models and our simulations to reflect these variants. And so we have been working for a number of months now on the Delta variant spike. And so we've upgraded, if you will, our whole virus models for the Delta variant. And this not only means that we've changed the spike protein, but there's also, our new virus also has the M proteins. There's hundreds of these M proteins tiled inside the membrane, and our model has those now, as well as the E proteins, or the E, which I think is a pentamer. It's an envelope protein, possibly an ion channel. Our model has that also. But so what I was very interested to do, so like you build this virus, we built this virus, but then like, what are we going to do with it? Well, in about 2017 or 2018, I had been recruited into an NSF center called CASE. And this stands for the Center for Aerosol Impacts on Chemistry of the Environment. And in this CASE center, in this NSF CASE center, what they're really wanting to try to do is understand sea spray aerosols. And so, it's one of these weird things where, like, I haven't actually talked very much about our work in sea spray aerosols because we started it and we're successful, but then we didn't actually publish it yet because the virus took over, and so it was just crazy. But so we had been working for years to develop all atom models of sea spray aerosols. And one of the big things where there's a lot of interest is trying to understand how the virus is surviving, or how it remains infectious in the aerosol phase. So we took our all atom model, which was part of the Gordon Bell Special Prize last year, and then we decided to try to actually put it into a respiratory aerosol, to build a respiratory aerosol, so that we could learn about what's happening inside the aerosol and what are the different conditions and features that of the air or of the virus itself that could impart this viability in the airborne phase. And so to do that, we really need to think about, or made us think about, where these particles are created. So in the deep lung, there are these in the lung itself. The lung is a really interesting, very cool environment because it's the air-water interface. So it's similar to sea spray in that sense. But basically, these cells have a bunch of things in them. They have surfactant, lung surfactant, which is basically shown here. It's got water, proteins, as well as lipids like DPPC, free floating lipids. And so this lung surfactant helps to keep the correct osmotic pressure and surface tension, of the air-water interface. These things, these particles also on these cells are these amazingly interesting molecules called mucins. Mucins are these large polymer, brush-like polymers that can be affected by, whose dynamics and so forth can be affected by concentrations of like calcium ions, as well as water content and pH. And so we actually built and also simulated some of the first all atom models of mucins. And so these are, again, you can see this got this long protein backbone and these O-linked glycans. They're like covered in O-linked glycans. So and then we basically took our all atom virus and our simulated deep lung fluid, which had all these different components in it, and we were able to create an all atom model of a respiratory aerosol. And so this guy is about 300 nanometers in diameter and has about 1 billion atoms, a little more than 1 billion atoms, and contains inside of it not only the virus, but all the other species that are there. And so what's cool about this is that it's, people have actually never seen before, because you can't see, because it's too difficult experimentally to image. We don't have the methods yet. These are giving people like the first views of what this virus may actually look like inside a respiratory aerosol particle. And I want to say that in addition to these like huge simulations, so we were able to actually just a few weeks ago, couple months now, we were able to build this and to actually get it to stably run on the Summit supercomputer. And it took up, the whole machine because it's definitely like pushing the limits. And, I think this systems like this will be used to develop the next generation exascale machines together with folks at Texas. But not only these huge simulations, which we can do, but I want to just reiterate that we can use this also as a platform for discovery where we're sort of switching the lens of the microscope and we can zoom in on these different sort of features, smaller parts of it to learn things. You know, not everything has to be a billion atoms. But while we have that, these are sort of this is really sort of a multi-scale platform for discovery. And we're looking at things like we're continuing to use the weighted ensemble technique along with HAMSM, this history augmented Markov state models to better extract kinetics from those data sets. And this is helping us look at how mutations, for example, in the Delta spike and now Omicron are actually changing the dynamics of the spike protein. We can also do things like look at how pH effects are impacting spike dynamics, because in these respiratory aerosols, I mean, it's amazing. You know, they drop pH because of various processes. So it'll be interesting to see like what's actually happening to the virus inside of these environments. Okay, and then I'll just say, you know, we were like just a couple of days ago or a week ago or so, Carl Zimmer wrote a second piece, sort of a follow-up on the study from last year, where in the New York Times, where he covered this, I think, in a really beautiful way. Sort of showcasing these different images also that have been developed in my group by these fantastic scientists I have in my team, and sort of, you know, helping to show people, you know, again, sort of like what the virus looks like in the aerosol. And this is really, we're so excited about sort of the future of, you know, at the intersection with experimental science now, and talking to so many different experimental collaborators about sort of testing the hypotheses coming out of this. And then, you know, so what is coming out of it? So, you know, those mucins that I was showing you, those are really interesting. And we think that those mucins basically are designed to protect cells that exist at the air-water interface. So one of our hypotheses is that these, as you can kind of see from this picture, is that these mucins may also be helping to protect the virus from the air-water interface, which is known to destroy molecular structure. So, you know, something is protecting it, and we think that might be part of it. And I'll just say there's some easy sort of low-hanging fruit hypotheses about charge interactions, where one of the things that we see, if you compare the original strain to the alpha strain to beta, delta, and now Omicron, you can see sort of the evolution of mutations, where in Omicron, well, it's a little bit hard to see, but Omicron has so many more mutations than any of the other ones. But each time we see a more transmissible strain, one of the features that we've noticed is that there's a significant uptick in the positive charge, the overall formal positive charge of the protein. That was true from alpha to delta, and that was true again from delta to Omicron. And one of our hypotheses is that, you know, it could be that these spike proteins, because they're taking on more positive formal charge, they're actually better able to interact with those things like mucins, which are negatively charged, as well as, so that could help protect them in flight during the aerosol phase, but it could also help the virus actually in the initial attachment to the host cells, because those glycosaminoglycans are also highly positively charged. So it could be a dual effect. In any case, those are experiments that are being run with collaborators like Ronit Freeman at the University of North Carolina. And I'll just say, sort of in closing, you know, we're really excited about trying to, again, sort of push the limits of simulation, and to try to learn something useful about, you know, what's happening to the virus, how it's working, and so forth. And there's a lot of questions that we can start to answer now when we have these tools that allow us to really sort of see things that until now had been sort of unimaginable. So yeah, and I'm really excited, and I hope to excite others about not only, you know, the work with viruses, but, you know, one can easily imagine how these types of simulations, data-centric, and, you know, really with so many really great methodological advancements, can really, what I like to call now in situ computational microscopy, because that's really what we're able to do. How that can help drive that virtuous cycle between theory and experiment to discover new things, not only for the virus, but also for diseases like cancer or neurodegenerative disease and so forth. Okay, and with that, I will definitely close, because I hope we have a little bit of time for questions. Sorry, I hope I didn't go too long. you know, there's just like so many people to thank. And the first Gordon Bell team of building the virus in 2020, there were about 30 authors and like 13 different institutions and 4 different supercomputing sites. And all those people are listed here. They're just so fantastic. And then this last year, we participated again in the Gordon Bell. We were finalists. We weren't winners. The Fugaku team had just a brilliant submission, and just they really did outstanding things. But this, you know, putting the virus in an aerosol and bringing that to bear with the AI simulation, multi-scale stuff, there were 50 people on that team, and even more institutions. And so just definitely too many people to thank. But yeah, I think that I just close by, you know, thanking you for your attention, and thanking all the folks at the NSF and the supercomputing sites, and you know, all over the world for just really sort of bringing their level best to try to learn something about this virus, and to do it quickly. And anyway, I've been so privileged to work with all these wonderful people. So I'm happy to take questions.
Thank you,
I hope that was
Rommie,
okay.
for a brilliant
Yeah.
talk. Let's give her a round of applause.